Hi, I'm Marianne Hawke. I'm a scientist with the San Diego Mycological Society. I love the way that mycology brings together such a diverse group of people who all have their own reasons for being interested in fungi, whether it's through science, cultivation, food, human health, ecological health, travel, or art. I'm really glad that you're here. So today we're going to dive into a fascinating topic, the mysterious hidden life of spores. You may not know it yet, but fungal spores are important in your life. These fascinating microscopic spores are colorful, beautiful, and incredibly complex, perfectly designed for the important role they play in nature. Let's dive in. When the microscope was invented around 1590, suddenly we saw a new world of living things in our water, in our food, and under our nose. The word spore was used for the first time by this man, Johann Hedwig, back in the 1700s. He was skilled at microscopy and biological illustration, and eventually he directed the Leipzig Botanical Garden. Hedwig was actually known as the father of bryology because of his work on mosses. So where do spores come from? Let's start with something simple. If you think of a typical mushroom that a child might draw, it has a cap on top and a stalk underneath. And then there are gills. That's where the spores are produced. These microscopic spores are designed to spread around through the air or by water in order to make even more mushrooms. Apparently, an 8-centimeter mushroom produces as many as 40 million spores in an hour. Of course, being science, it is a bit more complicated than that. There are 3 basic types of spores depending on how they're produced. Conidia, shown across the top, are vegetative spores, so they are asexual. The other 2 are sexual spores, and they're called basidiospores or ascospores, depending on the type of fungus that makes them. In the group of gilled mushrooms, the spore-forming tissues, called basidia, are located on the gills on the underside of the cap. The basidiospores are dropped from the gills when mature. In the other group, called sac fungi, the spore-producing sacs, called asci, are located on the inner surface of the mature fruiting body. These ascospores are released in a cloud when the asci break open. Ascospores, their development is shown here across the top, are produced by the sac fungi. So as their name implies, their spores are produced inside a sac. So they look a bit like peas in a pod, illustrated here by the fungus called Neurospora. Basidiospores, shown across the bottom, develop externally at the tip and are not enclosed in a sac. To help me remember the difference between them, I call ascospores innies and basidiospores outies. So now we know a bit more about different kinds of fungal spores and how they're produced. But what are they for? Basically, the answer is reproduction, dispersal, and survival. And of course, this being science, the different types of spores have a variety of specialized names depending on the type of fungus that produce them. But as it turns out, that's quite important for identifying fungi, and pathologists look at spore shape and appearance to tell them apart so they know which ones they're dealing with. Spores vary in shape from spherical to ellipsoid with different shaped ends. They also vary in their surface ornamentation, from warts and ridges to long pointed spines. Some spores, like the marine fungi, even have appendages around their middle or at their terminal ends. Ingoldian fungi. These aquatic fungi are named after C.T. Ingold, the British mycologist who first studied them. These are some examples of diverse spore morphologies that can be found in foam from freshwater streams. Who knew? Fungal spores can be complex and multi-layered. Here's what they can look like in cross-section. At the bottom is a picture I took with the scanning electron microscope showing the layers of the wall of a commonly found soil fungus. Spores are very beautiful, as this lovely illustration shows. They come in a dizzying array of shapes and sizes and look like they came from some kind of fantasy novel or alien world. When you look at them with an electron microscope, some fungal spores look a lot like plant pollen. That's pollen from a sunflower in the top left and a spore from puffball fungus in the lower right. Why does that matter? Because both can cause allergies in people. Just look at those menacing looking spikes. This is a picture of a variety of types of pollen taken with an electron microscope. Pollen that most commonly causes allergic reactions are produced by the plain-looking plants, trees, grasses, and weeds that do not have showy flowers. These plants manufacture small, light, dry pollen granules that are custom-made for wind transport, just like fungal spores. The pollen becomes lodged in the mucous membranes of the nasal passages when you breathe them in. The immune system produces immunoglobulin E, or IgE, antibodies to fight invading allergens. Even though everyone has some IgE, an allergic person has an unusually large army of these IgE defenders. Special cells called mast cells are frequently injured during this warring between the IgE antibodies and the allergens. When injured, the mast cells release a variety of chemicals into the tissues and blood, one of which is known as histamine. These chemicals are very irritating and cause itching, swelling, and fluid leaking from the cells. So that's why hay fever sufferers take antihistamines. Speaking of health, this article appeared in the LA Times just a few weeks ago. Scientists say that spores are likely spread on wind currents caused by wildfire. Wildland firefighters can get sick with valley fever after working on a fire incident. The illness is contracted by inhaling fungal spores of the genus Coccidioides, a pathogenic fungus that resides in the soil in certain parts of the southwestern United States. And it can cause fever, cough, and a rash. Here's another reason why fungal spores are important to people. Fungi can cause costly rot and diseases in our crops, such as wine grapes and tomatoes, or spoil the food in our fridge. San Diego County has a large flower growing and nursery industry that has to be protected from plant pathogens. Fungi can also cause problems for human health, from athlete's foot and yeast infections to lung infections like the valley fever I just mentioned, or blastomycosis. Spore dispersal. Fungi do produce a prolific amount of spores, and the way that these are dispersed in nature is very interesting. Some of the methods are active and some of them are passive. So what do we mean by that? Ballistospores are the active type that are usually shot from their basidia. The rusts, jelly fungi, and other groups of fungi may produce these kinds of active spores. So how do they shoot their spores? Well, one mechanism acts when a tiny droplet, called a Buller's drop, rapidly forms and shifts its mass in a way that pushes the spore. The energy of spore discharge comes from the rapid surface tension powered movement of the Buller's drop onto the spore surface that catapults the spore away. Ultra-high speed video cameras show that these ballistospores can catapult into the air at initial accelerations in excess of 10,000 G. That's pretty amazing. Coprophilous fungi use the dung of herbivores as substrates. The ascospores are sticky and darkly pigmented so they can survive gastric juices and UV light. The mechanisms of spore dispersal of these fungi guarantee that the spores are propelled from the dung onto nearby vegetation, mostly by these ballistic mechanisms, where the fungi will be eaten along with the plant material and pass through the animal digestive system to repeat the cycle. A fine example of an artillery fungus is Pilobolus, which lives in animal dung. It produces these black asexual sporangium that look like a cannonball on the end of a long stalk. Below the cannonball is a special balloon that fills with water in the morning sun. When the water pressure gets high enough, the balloon suddenly bursts, launching the cannonball into the air. Wikipedia says this involves acceleration equivalent to a human being launched at 100 times the speed of sound. That's pretty amazing. The orientation of the stalk towards the early morning sun apparently guarantees that the sporangium is shot some distance away, which improves the chance that it will attach to vegetation and be eaten by a new host. Start the cycle all over again. I love this one too. Isn't it pretty? Spirobola Stellatus, another type of cannonball fungus, develops basidiospores in a large gelatinous ball-like mass within this cup-shaped structure. Here's a cross-sectional view. At maturity, the inner layer of the cup separates from the outer layer and suddenly inverts like a trampoline, forcibly ejecting the spores containing the ball into the air. You just can't make this stuff up. Ascospores can be phototrophic too and move towards light, as in many members of the Pezizales. Explosive discharge is attributed to changes in turgor pressure that are caused by water uptake, sort of like a squirt gun. Then there's the passive type of spore dispersal that requires the help of wind, water, or animals. I love that our British friend Ingold called these methods blow off, splash off, or shake off, which kind of explains everything you need to know. These exquisite bird's nest fungi, so called because they look like tiny bird's nests with little eggs in it, are activated when hit by raindrops. Here's a cross-sectional view of a similar species. Isn't the level of complexity and sophistication remarkable for something less than the size of a penny? Now let's turn to spore color. You can set the cap of a mushroom down on a piece of paper and let it sit, and later you'll find fungal spores deposited in a ring pattern. This is called a spore print. The color of the spores can be used to help identify the fungus. Color in nature is another one of those fascinating topics that I could talk about for hours. While beautiful, the colors play a very important function. They are produced by secondary metabolism. That means there are chemicals not directly involved in normal growth, development, or reproduction of the organism, but instead they play important ecological functions. Some secondary compounds, such as pigments, polyols, and mycosporines, are associated with pathogenicity, and others with tolerance to environmental stressors, including temperature and UV light. The most famous fungal secondary metabolite you might have heard of is penicillin, which was discovered by Alexander Fleming in 1928. Spore coloration is often used as an aid to identification of species. Colors are generally pink, red, orange, yellow, purple, olive, green, or gray. The color may present itself in the spore wall. It could be within the cytoplasm, in oil droplets, or even in the membranes. Often the colors are caused by carotenoids such as the common orange beta-carotene or dark red lycopene. Other complex pigments like dark black melanin may be present. These are microscopic images of several different fungal spores. Agaricus spore walls are darkly pigmented. This is common in fungi found in xeric habitats. Lycoperdon spores appear brown, and spores of the desert fungus Bacteria are an orangey rust color. These colors are produced by the secondary metabolites such as anthocyanins. What's the role of color? Well, it provides protection from damaging effects of radiation like exposure to UV light and from desiccation. Coloration provides resistance during dispersal and aids long-term survival in harsh environments. The black melanins absorb radiation and dissipate energy into their double ring structure, which protects the spore membrane. Fungal pigments such as this dark brown one from Pizolithus can be extracted. They have a lot of uses, including the dyeing of wool and fabrics. At our annual fungus fair in Balboa Park each February, we usually have artisans who use fungal dyes to produce beautiful clothing products. Pigments can even be used as ink to make paintings and drawings. Fungal spores may also be fluorescent. I took these pictures of glowing spores with a special microscope under different wavelengths of light. So now we can finish off our talk by looking at some fun and interesting fungal spores. And I have to credit Dr. Pat Nolan of the San Diego Mycological Society for suggesting some of these. The conidia of this plant pathogen are quite distinctive and are known as three-tailed mice. Fusarium is commonly found in soil, and some types are important pathogens in plants and animals. It can have 3 different spore types. These ones, called macroconidia, are really big and look a bit like striped bananas. Some aquatic fungi have beautiful star shapes. These live in fresh water and eat leaves. These types of tetraradiate spores have even been found in fossils from 20 million years ago. Some, like these, look like cannonballs. Their dark coloration protects them from harsh environmental conditions. I'll end with this one, Spongiforma Squarepantsy Eye. Yes, it is named after the cartoon character SpongeBob SquarePants. I guess the scientists thought that when the spores were viewed under the scanning electron microscope, they looked like orange sea sponges. Isn't nature amazing? Thank you for listening to my talk, and I hope you enjoyed this glimpse into the hidden life of spores.